All right. Hello, everyone. Welcome to our Keys 2020 presentation week. We have heard some amazing presentations so far, and we are just so excited to hear the rest of the presentations that are given today to wrap up this amazing week. Right now, we are so excited to hear from Hailey and what she's participated in this summer. We do ask that you wait until the end of her presentation to ask any questions. Please make a note of them because we will have time set aside for questions and any type of comments or feedback you'd like to give. So without further ado, I'm going to turn it over to you, Hailey, and you can begin whenever you're ready.
Thank you. Hi, my name is Hailey Johnson, and this summer I worked in Dr. Bonnie LaFleur's lab. My lab works on biostatistical methods arranging data for biomarkers, which are indicators of diseases. Specifically, my research looked at gene expression as a biomarker of immune aging. This is my short abstract of my complete project, but I wanted to see how gene expression within older and younger donors related to something called immunosenescence, which is the deterioration of the immune system as we age. Using reproducible statistical methods in R, which is a statistical analysis software, we found patterns of expression between multiple genes and patterns of patient characteristics. So this is my complete poster from all of the steps of this summer, but I will be going through each section individually throughout this presentation. So first of all, there was a lot of background knowledge that I needed to have and that are necessary in order to understand my results. The primary organ of the immune system is called the thymus, and it actually produces T cells which help target and destroy foreign pathogens within our body. In the thymus, there's something called thymic epithelium, and these are cells and tissues within the thymus that actually mediate T cell development. They're extremely important to the entire function of the thymus. As we age, there's actually a phenomenon called Thymic Evolution, which our thymus area actually shrinks, as you can see from the figure on the right. Researchers still don't understand the mechanisms behind this, but we do know that it contributes to a susceptibility to illnesses, a poor response to vaccines, and also a higher chance of autoimmune diseases and cancers. The genetics in our body have a huge influence and importance on this process, from factors from thymic size, function, and the rate at which thymic involution occurs. We wanted to look at gene expression because of its importance. So the data that we had was derived from thymic tissue samples from a bunch of different patients. The data was recorded through the QuantiGene-Plex gene expression assay, which is conducted by the company Thermo Fisher Scientific, and they use branched DNA technology and signal amplification in order to actually detect the DNA and RNA transcripts. The tissue samples came from 35 total patients. 22 of the donors were under 18, and 13 of the donors were over 18. And this was really important information for us because we could affiliate gene expression values coming from an adult or a younger donor. The assay was also able to give us thymic areas. And by thymic areas, I'm referring to figure B, and this shows the thymic epithelium. And as said in the introduction, that's the healthy tissue that produces T cells for the thymus. So for our methods, there was a lot of data manipulation that we had to do, and it was all done in our studio. And if you look at this link below, and Robin's put it in the chat, if you click on that link, it shows all of the coding that I did this summer in order to get to my results. So every gene had something called a limit of detection, and this was just the lowest signal value that the assay could pick up. I'll be referring to the limit of detection as the LOD just to be more concise with my wording. So we started off by defining every value as above or below the limit of detection. So values were given a 1 if they were above the LOD and a 0 if they were below. And that way, when we calculated the means, it added up the ones and divided it by the values to give us a percent of values which were above the limit of detection. Going into our statistics, we had to create 2 separate groups to do an analysis on to accommodate for this limit of detection. The first group was genes with extreme patterns in expression percentages above the limit of detection. And we did this because if a gene had a really low percentage of values above the LOD, that means that there was very small expression in that group. And it's a very binary analysis for us to know whether there is high or low expression. The second group that we analyzed were genes with greater than 60% of their values above the LOD. And we used this because these values were properly detected by the assay, and they were accurate and precise measurements that we could do an analysis on. They weren't as obvious as the patterns in group 1, so this is where statistics came to help us. So our first group, which was the extremes, we found 2 separate genes that had very different patterns. The first was called ADIPOQ, and it had only 36.36% of values above the detection limit. And this tells us that there was a very small expression within the young donors, and it increased to 88.45% of values which were above the LOD in adults. So this is just telling us there is a huge increase in expression with age, whereas the opposite went for HPRT1. Only 86.36% of values were above the LOD in young, and only 34.62% were above the LOD in adults. And so this is showing us a decreasing expression with age. In our second group, which was those more obvious genes that had many of their values above the limit of detection, we found 18 different genes that were qualified for this group. But of those 18 genes, we only found 12 that were statistically significant. And when I'm talking about statistically significant, I'm talking about this p-value right here, which we were able to use to see if the changes and the differences were noteworthy, if they were obvious to us. If a p-value was less than 0.05, it tells us that there was a very significant, it tells us it was statistically significant. If you look at this top left average value right here, the averages for all the young donors were greater than the averages for the adults. And this was really interesting for us to look at, and we were able to put this into a visual representation called a beam plot. And the beam plot is basically just allowing us to see the density of expressions for each gene. The light blue represents the young donors, while the dark purple represents the adults. And this black line right here is just showing us the averages that you could see in the table. So for example, if we take VCAM1 and you take this mean value right here, we see that VCAM1 had an average value of 2.09 in young donors and 1.81 in adult donors. And our beam plot shows us the same thing. VCAM1 had an average value of a little over 2 for young donors and around what we could say is 1.8 for adult donors. We were also able to take patient characteristics, as mentioned in the introduction, and from our patient characteristics, we were specifically looking at the area of the thymic epithelium, which is that healthy thymus tissue. We were able to plot it on a graph, and we saw a really a high correlation between age and the percentage of thymic epithelium. As you can see by this line of best fit, the percentage of healthy cells within the thymus tissue sample decreased very rapidly with age. So what is this all telling us? It's telling us 19 out of the 20 genes that we were able to analyze had decreasing expression with age. And that was really surprising for us to find because we thought we would see fluctuating patterns of maybe some would have higher expression within adults, or maybe some would just stay the same, but they all had this similar pattern of a decreasing expression. So it is worthy to note that ADIPOQ from the first group did have increasing expression with age. The second thing we can talk about is the actual percentage of thymic epithelium. When we went into this project, we weren't sure if it was just the whole thymic area that was decreasing or whether the area of thymic epithelium was decreasing along with it. And this shows us that yes, there's a high correlation and it most definitely does. And this is probably a huge reason as to why there's a decline in T cell export with age. So moving forward, there's a lot of work that can be done with our data. Out of the 52 original genes we were studying, we were only able to properly analyze 20 because 32 of those genes had too many values under the limit of detection, and we couldn't use them because of their uncertainty. There was also a limitation for us with our sample size. If we look in our chart right here, these black dots are just showing us all 35 of these donors, and you can see there's a really high concentration of donors that were at age 0. And that tells us a lot of our thymic tissue samples came from infants and newborns. And if we're able to get more robust age distribution, that would be able to give our results more validation and credibility. These are some references that I had for my background knowledge. And this third link right here is actually to the assay, and it talks about the complex science and processes they did in the lab in order to get those gene expression values. This fourth link right here is just my HTML document I coded that shows all of the code I ran. I want to give a special thanks to Dr. Bonnie LaFleur. She's a brilliant statistician and an even more incredible person, and it was a really great experience to learn from her. Suraj Baraj and Jacob Song are the other two interns in my lab, and we really worked collaboratively this summer, and they made it a lot of fun for me. Robin Pratt, Brooke Moreno, and the key staff and crew for making all of this possible. This summer was definitely a challenging learning experience, but I couldn't have done it without them, and they offered me so much support. Thank you all for your time, and are there any questions?
Hi, Hailey.
Hi.
Do you happen to know or remember the R value, the correlation for your data?
Say that again?
The R value or the correlation for your data, do you happen to remember it?
No, we did not have an R value. We just dealt with the p-values and our raw data. We added code to actually put those p-values on the table.
On your scatter plot though, when you ran regression and you got your line of best fit?
No, we weren't actually able to add p-values, but in our next steps, if we had more time, I would love to add more p-values to that.
Awesome, thank you.
Hi, Hailey.
Hi, Mr. Thomas.
Hi. How is it working with R? Did you get pretty comfortable with that?
Oh my gosh, it was definitely a struggle at first. It was when we got into it, it was this really foreign thing for me. It was really scary at first, but when we started doing more manipulation and data wrangling, it got a lot more comfortable. And there's actually something called packages, which made the coding a lot easier for me. But it was definitely a very scary thing at first, but I was able to get a lot more comfortable, and I hope I'll be able to use R in my future.
Hey, Haley, I have a quick question. Great presentation, by the way. My question is, how has this internship and your experience this summer affected your thoughts on data science?
I think it's made me love data science a lot more. When I went into it, I didn't think I'd like it at all because I've never had any experience with computer science, and it was really hard for me at first. But after, the satisfaction of getting your code to run and seeing the pretty plots pop up and finding your results, I thought that was really fun for me, and to see the math along with it. So I think data science is a really interesting field, and I'd love to take more classes and learn more about it. But of course, I'm excited to be in the lab next year.
Hi, Haley. I have a question. Could you go back to the bean plots on the other slide?
Yeah.
So, what is that dotted line in the very middle mean?
So the line in the middle, that's just the average gene expression value for all of the genes. But it wasn't important for us to look at because we just wanted to see the differences between the genes themselves. But that's a great question.
Okay, thank you. Great presentation.
Thank you.
Hailey, did you all have any kind of speculation about what might have been the mechanism for the reduced expression as people age?
I did actually read a lot of, one of this article right here, Fox and One. Since we didn't get to be in the lab, we didn't get to work with the genes, just the statistics. But in this article right here, they actually talk about the methylation state. And they think that a reason that some of these genes, like Fox and One, that they studied might be decreasing with age, it might be because it's being hypermethylated. And that was really interesting to read about. But I hope in the lab I would be able to do more about why these are actually going on.
All right, if that concludes questions for Hailey, first we'd like to say congratulations, Hailey. Fantastic presentation. Second, we'd like to thank everyone for coming. Thank you so much for taking some time out of your Friday to come and support Hailey, to listen to the really cool research she contributed to and participated in. A big shout out to Dr. Bonnie LaFleur. Thank you so much for your mentorship this summer. This concludes your 3 student presentations, and each of them were phenomenal and unique, and that takes a lot of time on your behalf. So thank you so much for your mentorship this summer. At this time, we're going to ask Hailey to hang back so we can offer her additional feedback on her presentation. And thank you all so much for coming. Have a fantastic Friday.